Welcome to today's webinar, which is hosted by Hepatitis Delta Connect. My name is Beatrice Zovich, and I am a Public Health Program Manager at the Hepatitis B Foundation and the manager of our Hepatitis Delta Connect program. Thank you all so much for tuning in today. Today's webinar will consist of two parts. The first will be focused on research that was conducted at Karolinska Institutet in Stockholm, Sweden, to better understand the risk of liver cancer for those living with both hepatitis B and D viruses. The second part of the webinar will provide an overview of the Hepatitis B Foundation's US and international storytelling programs, Just Be and Be the Voice, through which we collect personal testimonials from those living with hepatitis B and D in order to decrease stigma, empower those affected by these viruses, and create change on a policy level. We hope that these presentations will inspire you to share your stories and get more involved in our work around both hepatitis B and Delta. Okay, so just a note about how to participate in the webinar today. So before we get started, just a reminder that all audience members are muted. We encourage you to use the chat feature to let us know who you are and where you are joining us from. If you have any technical challenges, please indicate your issue using the chat box and we will address it as soon as possible. If at any time during the presentation you have questions, you can feel free to enter them into the Q&A box. Questions will be addressed by the speakers at the conclusion of the presentation. This session is also being recorded and slide presentations will also be shared via email with all participants. So just a quick overview of our Hepatitis Delta Connect program. So Hep Delta Connect is a dedicated program of the Hepatitis B Foundation, and it was established in 2016 for the explicit purpose of raising awareness about hepatitis delta and promoting diagnosis, screening, research, and treatment, as well as providing support for those living with and affected by hepatitis delta. We do this through a variety of ways, including phone and email consults, social media, newsletters, a robust website, and webinars such as this one. We're hoping to continue to build and expand our programming throughout 2022, and we'll keep you posted about these developments and further ways and opportunities to get involved. So an introduction to today's speakers, I will introduce both at the same time. So our first speaker today is Habiba Kamal. She is a PhD student and researcher in the lab of So Aleman in the Division of Infectious Diseases and Dermatology at the Department of Medicine at Karolinska Institutet in Stockholm. And she will be sharing with us today her work on the paper Risk of Hepatocellular Carcinoma, or liver cancer, in hepatitis B and D virus co-infected patients, a systematic review and meta-analysis of longitudinal studies. And she'll also be giving us an overview of hepatitis itself. We are very excited to hear about her work, and we're very grateful to her for joining us today. Our second speaker will be Rhea Racho, who is a public policy and program manager at the Hepatitis B Foundation, and she will be providing us today with an overview of our two main storytelling programs, and will be able to answer any questions from anyone who might be interested in participating. So without further ado, I will invite Habiba to share her screen and get started. Thank you so much. I will stop sharing.
Thank you very much, Beatrice, for showing interest in my work and for having me today with you. Yes. So my name is Habiba Kamal. I'm a PhD student at Stockholm Research Lab at Infectious Diseases Department, Karolinska Institutet, Sweden, and I'm happy to present my work today before you. These are my disclosures. I think right now we know that Delta was discovered by viral hepatitis pioneer Rhea Racho in 1977 when he found a new agent in a series of patients presenting with severe fulminant hepatitis. And soon it was shown that the single-stranded RNA virus depending on B surface antigen for propagation and replication in the hepatocyte nucleus using host RNA polymerase resistant to antiviral therapy. And so far, 8 genotypes are discovered until now with difference in severity of disease course between different genotypes. Well, it is the unusual suspect in viral hepatitis. So it is the least common among viral hepatitis, less common than hepatitis C, hepatitis B, hepatitis E. The prevalence is still debated among several pooled analyses, with estimates saying it's 5 to 15% of chronic B carriers are living with chronic hepatitis delta. And unfortunately, no consensus among meta-analysis about the world prevalence so far. We have the least estimate around 12 million to 70 million, and meta-analysis were a little bit vigilant about including patients with chronic hepatitis delta and excluding those with fulminant disease, estimated that around 15 to 20 million persons are living with delta right now. And we can see like there are hot spots of viral delta, predominantly Mongolia having more than 60% of chronic B carriers are co-infected with delta. So the challenges in delta that low awareness about the disease, it's an orphan disease, no reflex screening so far, even in high income settings, and data coming from the USA are showing that low screening rates among chronic B carriers, even in USA. Certain discrepancy exists among the guidelines regarding screening for hepatitis delta. So while EASL and PUZZLE ask for screening for hepatitis delta among patients with chronic hepatitis B, a little bit ASLD, American Academy for Study of Liver Diseases, is unclear, and usually they look at hepatitis delta. It is a rare disease, and we should look for patients presenting with low replication of hepatitis B virus or coming from high endemic settings, rather than screening for all patients. Of course, not all patients are also screened for replication of the delta virus itself. So usually they stop at the antibodies and do not further go to the quantitative test for delta virus, which is an essential and important step also to identify patients at higher risk for liver-related outcomes, as I will explain. Through 40 years of studying hepatitis delta, cumulative evidence have shown that it has more aggressive course than patients with hepatitis B monoinfection alone. This was very evident in early patients, early studies in the 80s and the 90s. And usually those studies were involving patients with multiple comorbidities, poly blood transfusions, patients using intravenous drugs. And but this also was shown in later studies and even conducted in, and usually this aggressive course was shown in studies conducted in middle, high income settings. We lack studies conducted in endemic regions. And this quite also add to the challenge of studying hepatitis delta. We don't have data from endemic regions, longitudinal data, long-term follow-up of these patients, I can say. And here, based on a new meta-analysis that was published from Rhea Racho, and I am putting the references in the slides when you will get them. And of course, we are talking about super infected patients, and they will, further on, they will have a chronic course of this disease. Based on this meta-analysis, 30% will progress to liver cirrhosis within 5 years, and within 10 years, 15% will progress to hepatocellular carcinoma. Doing this review was really important for me because I went through the studies, and you can find that at baseline, at the time of delta diagnosis, you can see third of the patients to half of the patients have already evidence of liver cirrhosis at the start. And 2/3 of the patients have replicating RNA, Delta virus. And it's the persistence of Delta virus in those patients that was identified as a risk factor, as a predictor of worse disease course. And this is regardless of ethnicity or regardless of phenotype. New studies have shown that genotype 5, a new study from King's College, has shown that genotype 5 is usually in African patients from African descent, have better and favorable outcome. Of course, it is based on a small number of patients and needs further studying in large cohorts. So usually the concern when you find out that a patient has viral hepatitis, has chronic inflammation of the liver, is the liver cirrhosis. As always we say, like, the liver is a very silent organ. It doesn't manifest its decompensation until late. So it's very important to discover those patients early, patients with Delta or Delta, in my case, in my studies, and then further follow them for any signs of liver cirrhosis or decompensation. Predictors of liver decompensation in Delta are still ill-defined due to the low number of studied patients, but from now we can see that persistence of Delta RNA is one of these predictors of worse outcome. Certain genotypes were associated with worse outcome, genotype 1, genotype 3 in Amazonia. And also the usual suspects present in chronic liver diseases that might aggravate liver health, like presence of diabetes, obesity, NAFLD, metabolic syndrome. All of these contribute, alcohol overconsumption, of course, all of these contribute to worse outcome in those patients. While we are studying, we didn't find difference between males and females for progression into cirrhosis, but we know that hepatocellular carcinoma is more prevalent and incidence more in male patients rather than females. Older age usually is associated with increased risk of worse outcome. I think this is the main concern after discovering viral inflammation, the inflammation of the liver, and usually this is the preoccupation of the physician and the patient himself also, is how will I prevent occurrence of cancer? And I'm putting certain statistics about liver cancer, and we know that 90% of patients who develop liver cancer, it is on the background of liver cirrhosis, and 50% of today's liver cancer are attributed to B infection. So here we can look at the map of hepatocellular carcinoma all around the world, and hepatocellular carcinoma is the most predominant histological subtype of liver cancer. And we can see that it's not debated anymore that hepatitis B is an oncogenic virus, and it is identified by WHO as an oncogenic virus, together with hepatitis C, together with human papillomavirus, together with HIV, but hepatitis delta still not recognized as an oncogenic virus due to lack of evidence, of course. Hepatitis B, we know that it can occur in liver without cirrhosis, and fortunately, we have very good vaccination against hepatitis B that led to this massive vaccination programs that led to increase in the incidence of B and also increase in the incidence of hepatocellular carcinoma related to hepatitis B. And also there are effective treatments for hepatitis B. While these are not things that we can see right now in hepatitis delta. unfortunately. So I think this question for 4 decades, delta increases the risk of hepatocellular carcinoma or not? Because what was seen that it increases the decompensation and increases the mortality of patients due to the fulminant course, but whether it increases the risk of liver cancer was a little bit debated. Actually, the answer came with a leading meta-analysis from Dulci Alfayet, and she has done a massive analysis of 93 studies and looked at with broader criteria. In meta-analysis, you pool all the results of the research per the researcher criteria, and she has looked and she has seen that there is an increased risk, as it is mentioned in her paper. Several meta-analyses have came after and during the last 4 years. Several meta-analyses studying the prevalence of delta and also studying the risk of hepatocellular carcinoma in delta have came out. Well, the prevalence was debated, but the meta-analysis regarding the risk of hepatocellular carcinoma in delta were all consistent in there is increased risk of hepatocellular carcinoma in patients living with hepatitis delta than in patients living with B monoinfection alone. And this was my work, and thank you for being interested in it. I have different criteria of inclusion. I have only included longitudinal studies in this work. We thought like cross-sectional analysis will not give us the temporality of the risk for hepatocellular carcinoma in patients with delta. So we conducted 60,000 patients included among 12 studies spanning time duration from 1991 to 2019. And when we look closely to the populations included in this meta-analysis, we can see that delta patients, hepatitis B and D co-infected patients, the age they were younger, and this is consistent with the cohorts. Then we see usually patients with delta are younger than patients with hepatitis B monoinfection. Male proportions, they are similar, and liver cirrhosis is staggering. Patients with hepatitis delta, they usually present. In my 12 studies included per our priori in our analysis, around 60% were having cirrhosis at baseline, compared to 15% in patients with B monoinfection. And when we pooled the relative risk of the whole studies that are included, we have seen that delta infection poses twofold increased risk of hepatocellular carcinoma in co-infected patients, in patients who have delta, compared to patients with monoinfection. Let me tell you something about pooled analysis. Usually there are differences that we call the heterogeneity in between the studies, and it is, you cannot change that. Every study has its own setting, every study has its own follow-up period, every study has its own patients' characteristics, advanced liver diseases, patients taking treatment, other comorbidities among those patients. So when we try to adjust a little bit and restrict to cohorts that has adjusted estimates between populations with delta and populations with hepatitis B, we have seen that increased risk still present. And unfortunately, it did not correct the inherent heterogeneity in between the studies. Like I said before, there are limitations of the pooled analysis. And right now I'm talking about my work and also about the other meta-analysis that was published. Testing for delta RNA is not uniform among the studies, and we couldn't point out which patients with delta RNA are developing hepatocellular carcinoma. You cannot look individually to this during while you're doing the studies. And also a main limitation, there are missing information of patients on treatment among these studies. And this has contributed to significant heterogeneity, and also part of it, we could not see the genotypes, we could not study the genotypes, because genotypes usually are not mentioned in studies of Delta. Here I am sharing with you two recent analyses, more homogeneous cohorts, one from Italy and another from Taiwan, and both were in advanced liver diseases on treatment. And they have two cohorts, Delta cohort and the B cohort. So right now we are in front of patients with advanced liver disease and on NUC treatment. Still, Delta had increased risk, an additive increased risk for developing hepatocellular carcinoma, even among those two homogeneous cohorts. There are no studies describing the criteria, describing the characteristics of hepatocellular carcinoma in Delta. I find a study from 2012 from Abbas et al. It's in Pakistan, and in this large study, actually, this large number of hepatitis delta hepatocellular carcinoma, 92 patients from both groups, and he compared the characteristics between both groups. And delta patients tended to have more portal hypertension features, decreased liver size, patients having lower thrombocytes at the beginning, patients having larger varices on endoscopy, while patients with hepatitis B tended to have advanced HCC stages in this analysis with elevated alpha-fetoprotein. This study has certain limitations, conducted in a tertiary care center, and we could not look at further characteristics of those patients due to the transverse nature of this study. It's not a longitudinal study, it's a case control study. One of the things that we looked at in patients living with triple infection, HIV, hepatitis B, and hepatitis delta. And hepatitis delta has historic increased association with HIV patients. And we know that around 10% of people living with HIV are co-infected with hepatitis B also. And we found that there is a particular high risk for liver cancer among the patients with hepatitis delta and HIV also. And unfortunately, reflex testing is still missing in this important population. And when we looked here, and it was based, we are bound by the studies we are including in the meta-analysis. So we found that 2 studies included populations living with HIV, and we have found around 6 times fold to develop hepatocellular carcinoma among this group of patients. When we look to certain criteria that we have to look at when we are studying long-term risk, we call these criteria confounders. And those confounders, they can raise associations that might not be true. So certain confounders that certain studies might have, patients who are using intravenous drugs, patients who are having holy blood transfusions, and this was seen in the earlier studies, as I mentioned before, and also newer cohorts that tended to involve more populations from historical endemic regions, I can say, might have other comorbidities like hepatitis C, like parasitic liver diseases, and other environmental toxins like alpha toxin B1. And here I'm specifying areas in sub-Saharan Africa. Also, we could not look at the prevalence and influence of metabolic associated liver disease among those patients. And I think this is one of the main confounders in studies on hepatitis delta. When we look at, as I mentioned, it's the preoccupation of the physician as well as the preoccupation of the patient is hepatocellular carcinoma. And when we look at the hepatocellular carcinoma surveillance, where is delta from hepatocellular carcinoma surveillance? We can see at American Academy, unfortunately, there is no mentioning for hepatitis delta among high-risk group to be screened, no explicit mentioning. They mentioned liver cirrhosis in patients with liver cirrhosis, and we have to screen for liver cirrhosis, for hepatocellular carcinoma among the patients with liver cirrhosis. EASL guidelines, there is no explicit mentioning of hepatitis delta as an additive risk for hepatocellular carcinoma among chronic B patients. Part of the challenges in hepatitis delta is that the treatment for delta hasn't changed until recently, of course, for 30 years, and it was off-label use of interferon therapy. We all know that interferon therapy is, I might say, those who have seen hepatitis C treatment in the era of interferon therapy might resonate with me right now. It's a highly selective therapy. Not all patients can tolerate interferon therapy due to a lot of side effects. And also in hepatitis delta, it's a suboptimal virological response, around 20 to 30% answer for this treatment with delta RNA on detection, I can say. But functional cure is very rare to occur with, which is deletion or disappearance of hepatitis B surface antigen. The machinery for hepatitis delta to produce its virus does not occur. And this is with 48 weeks of subcutaneous interferon therapy. So interferon therapy until now is not the answer for hepatitis delta treatment. And of course, multiple studies have mentioned interferon in decreasing liver-related events, and has been shown that patients who are responding to interferon therapy have better disease course than those who are not responding. And I might tell you that, and it is well known, of course, that patients with cirrhosis, and we can see like 50% of patients with delta are diagnosed with cirrhosis at baseline, they are not eligible for interferon therapy. What about nuke therapy? Highly effective therapy in hepatitis B, entecavir, TAF, TDF are not, have been shown and have been extensively studied in patients with hepatitis delta, and they did not add further benefit to interferon therapy. And usually they are prescribed to patients late course of delta, while hepatitis B start to replicate again, and they are given usually to patients who are not eligible for interferon therapy in hepatitis. So I think the path forward for hepatitis delta, from my perspective as a PhD student working on delta since 4 years, that reflex testing is very important for all chronic hepatitis B carriers, that we need more prospective studies, more broad perspective on hepatitis delta long-term effects from regions of high endemicity, regions like Mongolia, regions like Sub-Saharan Africa, regions like the Amazonian, and of course, new therapies are nevertheless warranted for hepatitis delta. I would like to thank you, and I would like to thank patients living with hepatitis delta and their families and their caregivers. Of course, I would like to thank my PhD supervisors and my first co-author, Romina Fornes from Nelly Brusales Research Group, for conducting this work with me. And of course, the thanks extended to KI librarians for conducting the search for my paper. Thank you. I finished. Yes, thank you.
Thank you so much, Habiba. We really appreciate such a rich and insightful presentation. Thank you so much again. For audience members, if you have any questions, please feel free to put your questions in the Q&A box, and Habiba can answer them at the conclusion of both presentations. And I'd like to now invite Rhea to share her screen and get started with sharing about our storytelling programs. Thank you so much.
Great. Hi, everyone. So as Beatrice mentioned, I'm the Policy and Program Manager for the Hepatitis B Foundation. And one of my roles is managing our patient storytelling program. And so I'm going to start with describing an overview of our US-based storytelling program, Just B. And then I'll share one of our videos from the campaign, who's actually a person living with hepatitis delta. And then I'll also provide a quick overview of our newer Be the Voice International Storytelling Campaign. So Just Be was launched in 2017 in partnership with Story Center and the Association of Asian Pacific Community Health Organizations. This is a national and multilingual storytelling campaign that does share the true personal stories of people directly affected by hepatitis B and hepatitis delta. And we do this primarily through digital stories or videos that I'll show you, but also with accompanying printed materials and by engaging the storytellers in in-person events as well. The goals of the campaign are to increase awareness and advocacy around hepatitis B, to help decrease any hepatitis B related stigma or discrimination, to promote hepatitis B testing, vaccination, linkage to care and treatment, and to empower people living with chronic hepatitis B or hepatitis delta to share their stories, and again, to help educate the public and to inspire action. And so when I said digital stories, what do we mean by that? That is actually something that our partner, Story Center, has a methodology that they developed. And so since we partner with them to create the Just Be stories, we sort of follow their model for the, so they're a nonprofit organization that has been doing digital storytelling in public health, social justice settings in the US and globally. And so this is kind of an overview of their methodology. It blends different approaches from sharing oral history to doing education and using participatory methods. They take a community-based public health approach, and they use the stories, they share them widely as tools to help promote individual behavior change, community change, and policy change. And they are all, as you'll see in the videos, they're all first-person stories. They're created in a workshop process as a small group, and so it's not individual production, and it's also very hands-on. So storytellers during these workshops actually get to create their videos. And then, so building off of that a little bit, the workshops facilitated by Story Center and Hepatitis B Foundation staff, they're usually a 2 to 3 day weekend workshop, and it includes just kind of bringing people together first to get to know each other and hear their stories in what is called a story circle. And this is more, this is unscripted. This is just a lot of times it's people that are sharing their story maybe for the very first time, or at least, aspects of their story that they have never felt comfortable sharing. So we create a really supportive environment for people to feel open, to feel like they can share what they would like to share without being judged, but also with knowing that they're surrounded by people who have similar experiences and can relate to that. And then after that initial day of sharing stories, we also go into creating a script. So what people decide to share publicly in their videos may be different from what they share on the first day during a story circle. So we help participants refine a script for their video and do a little bit of training around effective storytelling, and then actually go into the hands-on process over the next couple of days of recording a voiceover for their video and gathering images, putting it all together, using a video editing platform to put it all together with the help of Story Center and other Hepatitis B Foundation staff. And what we found is that the process of creating the stories in this workshop environment is really important to the participants. So we do have the final video, or like the media product, that is able to help with education and advocacy. But for the participants themselves, it provides a space for sharing, for healing, and for community building, since hepatitis B can be a tough experience to talk about. So that's one thing that we love about bringing people together in person for the workshops, to create these videos. And then, so you have an idea now of what we're talking about. The videos, if you've never seen them, they're on our website at hepb.org/justb. And we're highlighting Joe today because he is actually, as of right now, of our 40 current storytellers, Joe is the only one who has a co-infection of B and Delta. And as you can see, he has a quote on here talking about participating in the only drug trial for Hep Delta at the time. And I'll just go ahead and share his video so that you can hear his story in his own words.
As a kid who loved nature, I was always smelling the flowers, digging in the garden. In the mid-70s, as a young gay flower child, I faced my fears and came out to myself, my mom, and friends. One day a friend said, "Hey Joe, your skin's a little yellow. You should see a doctor." I found out I have hepatitis B, but was told nothing about what that meant, and I just forgot about it. The AIDS crisis hit. Everybody was worried. I got tested and was relieved. I'm negative, but I did still have hepatitis B. I learned how to be safe. I did lots of research at hospital libraries. I faced my fears again, and I came out to my gay men's community about having hepatitis. I still didn't feel ill when, in 2013, my liver became inflamed. It turned out I'm also infected with a rare, serious virus, hepatitis Delta. In a follow-up email, the specialist typed, "There's no good treatment. Good luck." I wasn't about to leave it up to luck. I got online and found out about hepatitis Delta, and that it only affects people who also have hepatitis B. I wish they had had the Hep B vaccine back in the 70s. It could have prevented both viruses. So, the only drug trial for hepatitis Delta in the US was at the National Institutes of Health. I hoped this trial could save my life. I joined, flew across the country some 60 times in 6 years to get the treatments I needed. Last December, the Delta virus in my body was near zero. The treatment was really working. Then, with just 2 months of the trial left, I fell ill, really ill. I lost 25 pounds in 3 weeks, and my abdomen filled with fluid. In drug trials, the patient's well-being is always the first priority. My doctors took me off the drugs, and I've recovered. Sure, I'm disappointed I didn't finish the trial, but I feel so good about my part in the research. I'm still being tested, still monitoring myself, and I'm still hopeful we'll find a cure. Just be persistent.
Okay, sorry, I know I'm maybe blocking the message here a little bit, but Joe recorded that story in May in 2019, and he still works with us and volunteers with us on many activities, and is still really active in advocating for hepatitis delta research and hepatitis B and D cure research. So this is just an update on how he was doing as of last November. So I know he has many more updates today too. So just to round out the overview of the Just Be campaign, this slide, and you'll get a copy of these slides afterwards. So if you're interested in kind of seeing more and looking into the stories that we have, just wanted to highlight that we do have around 40 storytellers from 20 states in the US, and 18 of the videos are translated into different languages because many of our storytellers are bilingual. And we wanted to make sure stories are captured in multiple languages to be useful in different settings. And like I said, we engage with storytellers after the workshop to continue providing local and national storytelling or public speaking opportunities and leadership opportunities. This just shows how we promote the stories online and our website, social media mostly, but also at in-person events when we have them, we sometimes share the videos just like this, just to provide a personal perspective or invite the storyteller themselves to come and share their story and really be part of any meetings or advocacy awareness events. To be able to have people with lived experience at those events is really important. And so that's another way that the storytelling campaign has really helped us find people who are willing to do that and share their story. We have collected some great quotes from post-workshop surveys and interviews with storytellers. One that I wanted to highlight is that, or if you just look at kind of the keywords in these quotes, it's that people feel lonely at first when they get diagnosed, or they don't realize that there are other people that are going through similar struggles. And so again, bringing people together and then getting more stories out there in the public has helped hopefully started some momentum in getting more and more people to be open about their status and to find ways to use their stories to help encourage other people to take action. Which is what this quote highlights here, that their hope is that these stories can change how others, not just the people that are affected, but how people who aren't affected view those who are, and that, that more emphasis could be placed on finding treatment and cure for this condition. And then just for a couple of minutes, I wanted to share, so this Be the Voice Story Bank, as I mentioned, is newer. It has an international focus. And right now it's an online platform. We collect stories through a SurveyMonkey. So it's a form that people can go online and submit, whether they just want to write out their story in a narrative form, or there are questions that we have. There are prompts that you can either follow one by one, or you can just share your story. But the point is to collect written stories from around the world. And so we're starting, and then we collect contact information from people so that if they are interested in doing more, at least we have a basic idea of what their story and what their experience is and what they would like to share. And then our hope is to be able to publish those stories either as written stories, or to follow up with them to record similar either voice audio only recordings, or to create videos in the future. So right now we're working on just kind of collecting interest from people. And if you look at the form, again, you can provide as much information as you would like about different aspects of your experience, from diagnosis to treatment access challenges, to if you've ever experienced stigma or discrimination, or just how hepatitis B and Hep Delta have impacted your life. And so we are excited to be expanding this work, Be the Voice, not just the online story bank, but also actually collecting stories right now. Our colleague Catherine has collected stories from Nigeria, and we're working with a partner to collect stories from Taiwan. But this particular story bank is available to anyone around the world. And the links here for the story bank, this one will take you directly to the SurveyMonkey, but if you'd like to also stop and learn more first about the overall effort, I put a link to the blog announcing the original launch of the Be the Voice story bank. And then going back to the Just Be campaign, there's also a link. This is a different application process since Just Be requires an in-person workshop that participants attend. That's why that's focused only on US participants right now, but so the application is different, but it's open right now. And we actually, if anybody here is interested and in the US, we actually do have a workshop next month where we have a few spots open still. I know the timing is a bit short notice, but if anyone is interested, you can go on and apply right here or just email me directly to attend a workshop that we have in Berkeley, California next month. And so I just wanted to thank everybody, and I hope that this sparks any interest in sharing your story. And also, I know we have providers and community organizations on the webinar too, and we invite you to share the information about the storytelling programs with your communities, with people living with hepatitis. And also, if you have personal stories that you'd like to share about your experience working in the field, please feel free to contact us. Thank you.
Thank you so much, Rhea, for sharing that overview. And I think that's really great to hear, especially from Joe, and what he kind of went through to find a clinical trial really highlights the need for continued research and raised awareness of hepatitis delta. So certainly if anyone knows anyone or is experiencing hepatitis delta, we really encourage you to check out these programs. And so we will move to just some questions. So there was a question, very nice talk. Are there some parts of the world where there seems to be an increasing incidence or prevalence of hepatitis delta? And I think this is for Habiba. You want to speak to that?
Well, based on those who are screened right now, I might tell you this is not the true prevalence, I believe. There are areas of the West right now that's showing increase in the incidence of hepatitis delta, actually. And areas with the mass vaccination is exhibiting decrease in hepatitis delta. But due to the immigration from areas of high endemicity to areas of low endemicity, this observation is seen, that there is increase in the incidence of new cases, mostly among immigrants from endemic regions coming to USA, coming to West, North Europe, actually. South Europe, it was stable for a time, and it was decreasing and then stable due to the flux from immigrants coming from different areas of the world with high endemicity. And this, as much as it has posed certain challenge for healthcare in those countries, it has given an opportunity to study broader populations and for longer time, and to look at patients from other ethnicity other than Caucasian, to look at the outcome in those patients. So it has its good side into increasing the knowledge on hepatitis delta. So
great. And then there was also a question specifically about China. Not sure if you're aware, but do you know if there are any parts of China that have significant levels of hepatitis delta?
Well, I know that China right now, it's like 2 to 5% of chronic B carriers are co-infected, according to the latest meta-analysis. But I'm not fully aware of which parts of China.
Sure. So another question that came through, thank you for this presentation. The dialogue on hepatitis delta is very new in Canada. Yes, and this was a question about connecting patient advocacy groups. So yes, we can definitely we can definitely try to get in touch for sure. I'll put at the end of the presentation some info about getting in touch. And then the bulevirtide study is almost completed in the US and showing promising treatment. Yes, I believe bulevirtide. Habiba, do you want to share any thoughts on bulevirtide in general?
Bulevirtide is showing good results actually. Partly it's with interferon therapy is showing very good results, I can say. The issue with bulevirtide that there are relapse rates after finishing, but it's favoring, the nice thing with bulevirtide that recent analysis showed that it can be used in patients with cirrhosis also. They were ineligible to interferon therapy, and monotherapy, it's not only bulevirtide in patients with cirrhosis, was associated with good results. So I think bulevirtide is a very promising therapy for patients with delta.
Yeah, absolutely. That's very, 2022, I think, looks to be an exciting year. I know Gilead has submitted for approval to the FDA for bulevirtide or Hepcludex in the United States, and I know is hoping to expand that to other parts of the world as well. And I think there are 2 other promising drugs in clinical trials as well, and data will hopefully be available later this year. So, so that's very exciting. And I actually had a question. So I was wondering, Habiba, if you had thoughts on, so it seems like early detection and increased screening is a great way to kind of, catch hepatitis delta earlier. But it seems like there's kind of mixed, not everyone has standardized screening recommendations. And I was just wondering if there were thoughts on kind of what would need to happen to make reflex testing more available and screening recommendations more uniform, if you had any ideas.
Actually, I think it's very simple that once patient is diagnosed with hepatitis B, it can be like through computer or through the healthcare system automatically, like referral or with the laboratory to take an anti-delta antibody in this patient. And if it is positive, go forward for the quantitative RNA. Because I think people forget. Sometimes you forget about testing for other things, and it should be computed through be detected quickly. And certain studies right now showing that new patients with newly diagnosed hepatitis B are more screened than the relative pool of older cohorts of hepatitis B. So I think it's in the best case scenario, it's automated reflex screening for patients with hepatitis B for delta. Certain things also it's deficient in the guidelines. When you meet a patient with hepatitis B with all the concerns, you start to tell him about his family members, about sexual partners, and you start to tell him, okay, you can test for hepatitis B. While he can come or she can come to you again and tell you, oh, I found out that my partner is having hepatitis B. We don't go further to test for delta also. So it might be also good to extend those screening for family members, and family members is a very well-known transmission route for patients with delta. I have found it's part of delta patients associated also with transfamilial transmission. So it's all about awareness about the hepatitis delta and automated reflex screening.
Absolutely. Yeah, I think that's so important, and it's still such an kind of represented disease. So those are definitely great recommendations, and hopefully those can become formalized as time goes on and delta continues to become more of the public profile and conversation. Great. Well, thank you so much again. I'll give just one more minute if anyone else has any questions. And I will also share my screen for our final slide. Okay, so I just wanted to put this information up here. This is our contact information, and I wanted to thank Habiba and Rhea so much again for your time and for sharing all of this really important information. And thank you so much to everyone for participating today. We really hope that you found this presentation to be informative and valuable for your work or your life, and hope that you can apply some of this information moving forward. And if you are living with hepatitis delta, or if you know of anyone who might be interested in sharing their story, that's a really great way to help to raise the profile and build awareness. And personal stories and testimonials really go so far in terms of changing minds and just raising consciousness. So please do feel free to share our Be the Voice and Just Be storytelling programs. And we encourage you to complete the evaluation that will after the webinar concludes. And we'll also share a recording of this webinar and the slide presentations out to all attendees via email. And we also encourage you to sign up. We have a newsletter list for hepatitis delta. One is for providers and one is for people living with and affected by hep delta. So those links to sign up for the mailing list will be put in the chat, and we encourage you to sign up so that you can receive updates about hepatitis delta drug development, clinical trials, research, and other news. And again, please do feel free. I've put our contact information on the slide, and it's also in the chat. You can visit our website. We have a contact us form on there, and you can also feel free to reach out to this email and get in touch with us. We would love to, to partner and build advocacy with organizations around the world. So please do feel free to reach out. And also, if there are any questions that you didn't get a chance to answer, you can email those to us as well, or that you didn't get a chance to ask. Great.
Well, I can see Amin Uriurdi is asking the two other clinical trial drugs. Well, there are two clinical drugs actually in type of interferon. It's called interferon lambda, and it's more specific working on hepatocytes. So it has less side effects with conventional interferon therapy. It's still under trial, and also there is prenylation. It's a drug called lonafarnib, and it is targeting a key step for delta variant assembly, and it is showing also promising results, but it's still in phase 2 trials. I can say phase 2, phase 3 trials. I can see. So I hope I answered you.
Yes, thank you so much for the question, and thank you, Habiba, for catching that. Yeah, so we hope, do stay in touch with us, and we hope, this year the landscape seems to be changing for hepatitis delta. So hopefully these new clinical trials and new drugs will help people become more aware, and will hopefully prompt new screening recommendations and more standardized screening recommendations around the world as well, so that we can catch more of the virus and treat it early. So. All right, well, thank you so much again to our speakers, and thank you to everyone for joining us today. And we hope you have a wonderful rest of the day. All right, take care. Thank you.